Thank you very much. My name is Peter Fonagy, and I have the privilege of chairing this afternoon's lecture by Professor Goodyer. I think most of us know of Professor Goodyer if we do not have the privilege of knowing him. To introduce him just briefly, he is Foundation Professor of Child and Adolescent Psychiatry at the University of Cambridge. Also, since 1999, he has been a Fellow of the Academy of Medical Sciences of the United Kingdom, which is a high privilege. His research, that most of us know, has brought together life events, hormones, and more recently its interaction of hormones and life events at the level of the brain. He has done a number of important longitudinal studies, including genetic studies. He is one of the key scientists, clinicians in the world doing research on depression at the moment, particularly his work on the role of the HP axis and cognitive functions in emotion regulation and mood disorders has been absolutely pivotal. He has a recent book on the origins of common mental disorders. He has a number of recent papers, including an absolutely essential paper for everyone to read in the British Medical Journal on the results of the ADEPT trial of depression that he co-led with the late Dick Harrington. He is a man for acronyms. He leads the CHEMP project, which is the Cambridge Hormones and Mood Project, the ROOTS project, investigating the origins of adolescent psychopathology, the People Project, which is a family study, and of course the ADEPT project that's reporting currently the adolescent depression, antidepressants and psychotherapy study. In terms of getting to know Ian personally, he is probably one of the sharpest intellects in child psychiatry at the moment. He knows an unbelievable amount, and he's able to, what's even more remarkable, retrieve this information at the drop of a hat. So I know for certain that we will have a great deal to learn this afternoon about unipolar depression in young people, mechanisms and processes in the onset and course of the disorder. Please join me in welcoming Ian Goodyer.
Thank you. I do hate being introduced. Thank you, Peter. The generic title for this conference is Bridging the Gaps, and so I thought to myself, how can we certainly no trouble finding gaps. It's child psychiatry, there's more gaps than bridges. But I thought we might talk about what bridges we might be building. And so that's what I'm going to do, and I'm going to use some follow-up studies that we've done, plus some of the neuroscience that others have done so well as proof of principle studies, and a bit of genetics. It was very kind of Peter to mention all our studies. Almost all of those studies are unpublished, which makes it rather difficult for you to consider them in their quality. First of all, let me just set the context, and I want to confine my remarks to this notion of major unipolar depression, which has this phenomenological, categorical and descriptive set of characteristics. And I think one of the things about the conference that is so exciting, and I hope we'll see as conferences throughout the world pursue over the next decade, that this concept is going to change. We're going to think about mood dysregulation in a much broader and I hope more neuroscientific manner. We're going to think about other forms of mood disturbances besides this one that has served us well, but like all good classifications will eventually have to give way. But for this evening, this is fundamentally what I'm talking about. The adolescents between the age of 11 and 18 who present with a mood state and who have other symptoms that can be elicited at a clinical interview, and that these can be gathered together in the presence of impairment that has to be sufficient to be diagnosed as major unipolar depressive episode. Also, just for clarification, almost all of the work that I'm going to be talking about, mine or other people, is on quite ill individuals, it seems to me. So the notion of how this generalizes to cohort and studies that have been published just requires a pause, because quite a lot of these findings are not from unselected cohort samples. So we could start with a summary slide, which is that one of the things about this condition is that it has a very high risk of recurrence. So from the point of the first episode, anywhere between 11 and 17, the chances of having a second episode over the next 5 years is, roughly speaking, somewhere between 40 and 50%. There's another 30% or so from epidemiological studies, the Smoky Mountain study and the Dunedin study, Christchurch study, USA's comorbidity studies, where the concept of impairments, if taken as the outcome, will show an even greater and more widespread fundamental problem. So you may be depressed in adolescence, but you may be impaired as an adult, even if you don't meet diagnostic criteria as an adult. You may have depression as an adolescent and have recurrent depressions into adolescence. You may be impaired in adolescence and childhood, not meet diagnostic criteria, but meet diagnostic criteria later, or all of the above. One of the most difficult issues that I won't dwell on because I don't think we know enough about it are individuals who sort of have something. And to sort of have something between 11 and 17, to have 4 symptoms of depression, to have moderate impairment with 3 symptoms of depression, there are now 2 or 3 studies, cohort, sequential, longitudinal design studies, that show that's not a particularly good thing either. But it's a problem to say that. It's a problem in all sorts of ways. And I shan't be addressing that issue, which is of significance for in this talk. I want to share with you something. Let me just go back. One of the conceptual issues about this is that studies in the state, Danny Pine in the States, Eric von Bonn, formerly at the Maudsley, Dick Harrington, and others, have all confirmed this recurrent risk. But something that happened in a set of our clinical studies that troubled me was that I was beginning to get the distinct impression that recurrent risk is one thing, but that there was a group of depressed patients who seemed to me to never get better at all. This was an anecdote from some longitudinal studies that I'm not going to talk about. But I thought that we would test that by doing a follow-up study, which we couldn't do under the new ethical rules in the UK. We followed up every depressed child that we'd seen in the 80s and 90s, about 125. And just to summarise, that we had a group who'd been in our clinical studies and a group who'd been in our community studies. The community study group had never been treated, and that's one of the things we wouldn't be allowed to do today. They had been recognised as undetected depressive episodes, and the clinically depressed ones had been treated. And no surprise really that at a 9-year follow-up, so they're into their 20s and median age was 24, that again you have a substantial proportion of this small sample, intensively studied as adolescents from a variety of ends, who show subsequent depression, much more than the community subject that was a periodic follow-up group. So these were seen at the same period in time, and they were followed up over that period. But what really confirmed my worries, and was, I have to say, somewhat despondent for the research assistants, was that we did indeed have a division in this group of disorders into adult life, in which 22%, which is quite a lot, never got better at all. They were depressed in adolescence, they were treated, and they remained fundamentally depressed. And the follow-up revealed all the sorts of things that those of you who practice will have heard but really don't want to hear. These were the ones who were treatment resistant. They did not like coming to the clinic. They were reluctant attenders. They were non-compliant in certain respects. They had a variety of features where you wanted to say, don't you realise that this is a bad thing to have? If only you do a bit better for yourself, maybe your outcomes would be better. We didn't say those things then. So persistently depressed individuals and recurrent episodes, and were these distinct groups. And a quick look, and using a few fairly standard multivariate tests on a small sample, showed four things that were rather interesting. Recurrent groups were more females than males, but persistent were more males than females. Very small samples, taken with a pinch of salt, proof of principles. Prior duration had really no effect on the risk for recurrence over the next decade, but the longer you'd been ill, the more likely you were to be persistent. Similarly with severity, these two things were correlated, and a previous non-depressive child psychiatric history. So a picture that won't surprise clinical practitioners who've been in this business for a while. Males with a long and severe duration of disorder, who had previous histories of a variety of anxiety and behavioural disorders, probably known to services, were much more likely to be in the persistent group. Females, no particular duration, perhaps showing more heterogeneity, same level of severity, but did not have a prior child psychiatric history, were more likely to be in the recurrent group. Doesn't look like it's a good thing to be depressed. Adolescent depression is bad for adult life. And persistent as well as recurrent illness is possible, and substance misuse turned out to be one of the likely correlates of persistence, and that won't surprise many of my American friends who have been seeing this for a while. And we were struck by the very rapid and extremely impairing use of marijuana, mostly in this group who persisted. But it does raise an interesting set of questions about differential consequences for outcome for males and females, which is somewhat different for the differential consequences for onset for males and females. If we were going to start doing sex-differentiated studies, and I think we probably all will, we might have to think about the way in which the substrate on which we work, the brain, is sex-differentiated in particular ways as a substrate level, and then is subsequently acted upon differentially because of events, including treating depression as an event for the brain, to result in differential pathways of neurodevelopment. And the next bit of this lecture is trying to think about, if that was even marginally true, where would we go with the notion of looking at the brain and thinking about the way in which experiences in the world and the brain might be talking to each other to upload and download risks, not only for depression, but for the consequences of having a depressive episode. We took a little group of these children. This is again a proof of principle study. I just give it to you as a kind of invitation to think about these things. These are not definitive findings. But the question was, what do the brains of adults who had been depressed in adolescence look like? And Ruth Hewson, a doctoral student who was with us, did a smart thing. She took some of our follow-up studies from clients from the study that I told you about, who had been depressed but had not had a recurrence and had been perfectly well for at least 2 years. And she found 13 such individuals in the cohort, and we matched them with 12 controls who'd never been depressed or had a mental illness in their life. And we did two things. We did some structural imaging, but we also did some functional tasks, just to see if we could answer the question, would these individuals who would not be picked out from the street, these are going to be entirely well young adults, would they reveal any differences at a structural or a functional level? And we chose to look at memory because of a particular hypothesis that was growing, which was that corticosteroids and the dentate gyres of the hippocampus were playing around with each other in a way that might not be too healthy if steroid levels were high. This was an animal study, animal work that had been around for a decade. We'd just done some work ourselves in our rodent lab, and others were publishing extensively. So we thought that we'd look at memory. So this was a region of interest, hypothesis-driven pilot project to see if we could do the tasks, if we could test the hypothesis, and whether or not there was, in general, a reason to believe that these individuals' brains might be different in some way. Now, we established that there were no current offline neurocognitive differences between the groups, and there were no current psychosocial or mental state differences between the groups. So, as I say, they were completely normal. And if you look at these findings very quickly, I've always found it's easy to look, not quite so clear to understand the blobs. But effectively, in the controls, deep to shallow encoding, which is essentially emotionally charged words against fairly neutral words, causes activity in the left and right inferior frontal gyri and the right hippocampus in the controls, but exactly the same functional task in the participants who, just to remind you, are completely well, were depressed as adolescents. They show generally increased activation in the prefrontal cortex, with little corresponding increase in hippocampal region. Perfectly well, functioning normally, different parts of the brain lighting up under same task circumstances. We did something secondary, which is a retrieval task, and here, in the retrieval task, the depressives appeared almost as if it were to be working harder. One has to be careful with that phrase. There was greater activity in the hippocampal regions only in those who'd been depressed. So this very, very preliminary study, which was in the general realm of a number of studies on depressed patients who'd been followed up and retested in the imaging paradigms, showed that we had a difference in the neural networks observing memory functions in adults with a history of adolescent depression. Nothing wrong with them, they just had a different circuit, or at least they had a different threshold, or they had some kind of integral difference in the way in which that circuit was operating under that test at that time. There seemed to be hyperactivation of the prefrontal cortex at encoding in order to achieve the behavioural equivalent results. Could this mean different brains, same results? Well, we were playing around with that. Of course, our extremely busy friends in the United States were well ahead of us. And they'd been looking, using a number of things, at serotonin and emotion using neuroimaging. Now, you probably know that from a developmental biology perspective, serotonin is a really key index of brain development. It's a fascinating, in the fetal brain and in the early perinatal brain, serotonin's pretty important stuff. Quite how the signalling molecules work is a matter of great activity, but they have this developmental role. This is not about psychopathology, this is about architecture and organisation of neural systems and possibly neural networks and maps. So there's an influence on neural systems that subserve emotion recognition in particular, and there's a key importance of having a competent serotonin system as a chemical indices of central neuroaffective integrity. So these things have been underpinning and are underpinning lots of studies, as you know. This is one of the nice complicated cartoons of serotonin, and this is this wonderful promoter that has given so many people a career and will continue to do so for a bit, I'm sure, because it's got a fantastic kind of bridge, to use a metaphor, between this very complicated set of post-receptor sites and all this chemistry in here. And it's been really remarkably good for studies. It gets a bit more tricky when you want to study things over here, because there's 7 of these, each has got at least 3 subunits, they've all got second messages, and it just gets more tricky, and it will. And you could include this kind of model in getting it trickier, and we heard Judy Rappaport talk about CNVs are going to come on board. The genetics of this is going to become fascinating, but very tricky, and certainly not something we're going to work out shortly. So this shorthand for the promoter has been remarkably helpful. And the genotype frequencies of these are large enough to allow lots of studies, and there are lots, and they continue, and we're doing them too. And here's one about the brain, which kind of helps the little story that we're trying to build. This is from, I think, from Danny Pine's group in the NIMH, Robertson Ney and colleagues, in which they looked at S allele carriers, just to remind you that you can be a homozygous for the S. This is the short arm of the serotonin transporter, which a more inefficient gene to have from the point of view of serotonin operating. You can be heterozygote and you can be homozygote. And because there's enough, as it were, without getting too clever, you can get a sample size and you can start doing some work. And Robertson and colleagues have done this work and shown a volume reduction in S allele carriers in the limbic cortex and in the amygdala. And these grey matter volume reductions, without going into what this might mean, if you recall, given that serotonin's key in neurodevelopment, this might well be an extremely interesting beginning to understanding how the genetics of the brain influences the substrate upon which subsequent life experiences work. Life experiences can be seen as social, but remember that for a geneticist, and I'm not one, by and large everything that's not genetic is the environment. So it's not just about social environments. There are a wide range of environments from the cellular upwards that might be important. But here's a study suggesting that there are indeed brain-based differences as a consequence of relatively simple dissociation on one genotype. And this extremely important study that I thought was beautifully done by Hariri and colleagues in Pittsburgh, I believe, shows that not only is it about volume reduction, but if you have an S carrier, then you're going to be sensitive to an emotion task. Bilaterally in the amygdala, S carriers, homozygote and heterozygotes, showing increased activity on the bold signal in the presence of a fearful facial expression. So there's us doing a little study on memory and looking at individuals who've been depressed. I'm going to try and bridge this gap in the next few slides. And here's people doing studies showing that the S allele is got something to do perhaps with neuroarchitecture and also perhaps with functional changes in areas of emotion. And here's a part of the Robertson-Ney study group which shows also a memory task, which is about remembering and forgetting faces. And our memory task is about words and the emotional content of words, and the difference between coding and encoding words with emotional meaning. And this is about looking at something. And again, they've got activity in the amygdala and the anterior hippocampus, and these are depressed individuals. And this is this activity shaped up in a bar graph. Goes up in depression. Just to remind you that I'm making a bit of a leap across this bridge, but the activity in the bold interface in our study went up in depression too, in a memory task on words. And they show that there doesn't seem to be much happening for anxiety. That was rather interesting, I thought, and the controls. So quite different groups and quite different things and quite different questions, beginning without even talking to each other, as far as I know, beginning to address questions that are focusing in on genetically sensitive structural and functional neuroimaging in individuals, some of whom actually have something, depression, some of whom have had something, depression, and yet beginning to find, as it were, a kind of broad area of similarity. Structure and function of emotion centers in the brain are serotonin sensitive. Lower volumes appear to be found with higher activity as a general principle. and the serotonin promoter gene seems to index these individuals rather nicely, allowing many more studies that are either in the journals, on their way to the journals, coming out the other end of the journals. What's that got to do with the longitudinal study that I showed you, or the studies by von Bonn and Harrington and Danny Pine and many others, and particularly this wonderful sequential cohort analysis in New Zealand from both Dunedin and Christchurch? Well, of course, we need to know if being an S, let me suggest for shorthand, S brain, makes a difference to, for example, the recent observation by Terry Moffat and Avi Caspi of the sequential correlational relationship between anxiety at one stage of life and depression at another, in either direction. Or the finding from David Ferguson in New Zealand that minor depression has a very high rate of major depression over the next 10 years, but if we knew what the genotype was, would that help us understand who was more likely or not? And if we neuroimage them, would that help us understand those things? Those are questions, it's not that they forgot to do those questions, they're the questions that arise and are things that can be done. Would we have a greater handle on mechanisms? Because the mechanism that's being proposed here is that a regulatory process that involves a stimulus that has an emotional connection is going to be differentially managed in individuals with different genotypes in the emotion centres of the brain. That's what the mechanism would be about. So S allele carriers may have different brain structures, also the substrate upon which environments work, or the early life course environments may exert effects on the S brain, which might be sensitized to those environments, that might bring about differences in structures and functions. The interplay here being between genes and neurons and the environment. However, it does trouble me when I read through, and I've read about 50 to 100 of these papers in the last few months, I suddenly thought we must remind everyone, including myself, just because you carry a gene doesn't make you a bad person. This isn't a bad gene. This gene isn't causing disease. It's one little polymorphism in an array of over 3,000 neuroactive genes. Very careful here. So I just thought I'd remind myself and you that it isn't a bad thing. It's an interesting thing. But it's interesting in a way that has been capitalized on by the epidemiologists doing gene-environment interaction studies. So if we come out of the brain but keep the genotype and we look at the environment, this terribly important conceptual paper and empirical finding by Avi Caspi and Terry Moffat on their Dunedin study, that there's a gene-environment interaction. I'm sure you've all seen this, but just to remind you, there's a bunch of stressful life events. I will come back to this in a sec. Over a 5-year period, over quintile, that's about one a year. It's about what you would expect. And here are the 3 types: those with the homozygous for the S, and they're heterozygotes, and these are the homozygous for the L. And this is the number of depressive symptoms. And you can see quite clearly that there's a gene-by-environment interaction for depressive symptoms. It's biggest in the homozygotes. It's there in the SLs, quite different from the Ls. A whole series of things came out in that paper about discussing what this might mean. Findings weren't overwhelming, but they were unique and novel and very, very important, and replicated. They were replicated by a group that sat 2 doors away from them in the same institution on a entirely different cohort. Slightly differently, I have to say, in that the really big difference was in the homozygotes. This is a depression score. There's the genotype. The adversity index was different. I will come back to that in a sec. And their finding was confined to females. So just to do that again, this is not sex differentiated. It's about major life events, and it's about a gene by major life event interaction with that particular genotype. This is a much more composite environmental risk that isn't exploded in the diagram, but is about chronic adversities. That could be, we could spend the rest of this talk talking about what that meant. And it looks like the homozygous findings are particularly noticeable. And then Joan Kaufman's paper, in which maltreated children, again a rather composite notion of maltreatment, with higher depression scores, and sorry that's not marked properly, there's the genotype. This is particularly true if the children lack social supports as measured by Joan. So that there's a series of papers, and Ken Kendler has another one, and there are more, Savilli recently in molecular psychiatry, and apologies to anyone in this room who's also got one coming that I haven't talked about. There are over 20 replications. Not all replications, there are some non-replications too, but by and large, pretty robust findings in which there's a gene by E environment in which the S allele is somehow indexing something at the level of sensitivity, I think might be the easiest and also the most imprecise way to put it, and that environments are coming down and somehow not through correlates. It's the argument is this is not a person-dependent environmental correlate, not a gene-environment correlation, and that's certainly being hotly debated by some, but that we've got this S-brain, we've got a gene-by-environment interaction, and we've got depression. Just a few points. You must remember that one gene may exert effects on the same environmental measure. More than one gene is likely to do that. Robust measures of the environment are terribly important, because if you have low reliability for your environmental measures, you don't get very strong findings. So one of the reasons why people are getting their environmental measures differential in their studies is they're looking within their own cohorts for the best, the strongest environmental measure. Major life events. In the Virginia twin study, it was interesting to see that slightly milder life events were rather important. In the Tagliaini study, some kind of high index of risk with a composite of the events, adversities, chronic difficulties. In Joan Kaufman's study, we have maltreatment index plus low social supports. But they all seem to be saying there's something important about the gene, and then we've got all these differential kinds of environments. Maybe one of the steps we have to do is to firm up our dissection of the environment, whilst we're getting very excited also about the genes. So let me move on to you can just hang on to those set of thoughts for a bit, and let's move back to something that Peter said that we've done a lot of, which was cortisol and the emotion experience, because cortisol is a peripheral chemical marker of emotion learning. Animal data for decades on how you can really upset the poor little rodents when you mess around with corticosteroid levels and you give them tasks to do or not to do. They have a rapid rise in cortisol, demarcates encoding of spatial objects and also objects of valence, and if you interfere with that rapid rise, they don't encode properly and they have trouble remembering stuff. Cortisol in the periphery, but it's regulated at the level of the hippocampus, and it's the dentate gyrus and other components around that area, bits in the amygdala and the prefrontal cortex, and a whole array of neuroanatomy that gets called into play when you're faced with a novel stimulus, and that's true for us humans too. And cortisol levels are highly sensitive to these environmental demands, as you know. I'm not going to talk about a really big piece of work. I was going to, but we'll be here forever, which is from Sonia Lupin's group in Montreal, which I think is terribly important because she's done the best, for me, the best work on being able to do things we can't do in kids, which is mess about chemically with cortisol in intact human beings and demonstrate memory deficits. And she also can demonstrate that she can take them away with anti-glucocorticoid medications, providing us with a quasi-experimental validity for the relationship between the HPA axis and mnemonic function. But one of the key groups, also from Montreal, Michael Mina's group, has developed this model, which they continue to work on. This is, again, apologies, again, anyone in the room who knows the work well. There have been more recent papers with some more molecular biology, but here's an epigenetic model in which maternal experience exerts effects on the rodent brain. This is environments influencing genes. And lo and behold, that in this model is our important friend serotonin. But also in this model is our friend glucocorticoids. And in between a whole lot of other stuff, particularly trophic genes, which I'm not going to talk about for the sake of time. In a nutshell, what this group have demonstrated beautifully, because they've done some reverse experiments too, is that if you interfere with maternal experience, you interfere with the brain of the infant. If you interfere with the brain of the infant in an adverse way, because the maternal experiences are negative, you will produce permanent changes in the HPA axis via a system of chemical change in the brain that involves serotonin and methylation of glucocorticoid receptor areas. You don't want histones around your GR. It's not good for you. I haven't ever said that to a patient, but perhaps one day I will, if I have enough evidence. So let me take you away from this extraordinarily important piece of work, but keep the concept of early maternal experiences being negative for the child, but negative for that child's brain and that HPA axis. If that were true, then a longitudinal study of infants exposed to something undesirable, like poor maternal care, should produce individual differences in the HPA axis over time. And we were fortunate enough to be in a position to test that, because for 20 years, Lynn Murray, Peter Cooper, and Sarah Halligan, who joined the group later, and Joe Herbert and myself, but particularly Lynn and Peter, have been driving forward a study which is about child development. And I just hasten to say that this is where you take advantage, if you're fortunate, of a cohort that had an entirely different question asked of it in 1985. It had nothing to do with the HPA axis or depression. It was about the formation of social cognitive structures in infants who were exposed to poor maternal care. And, we had at that time, none of this was available to us, but we've managed to, as it were, piggyback on this child development study. And these are a very small but intensively studied set of children who are now 20, who had been followed at 6 months and 18 months and 5 years and 7 and 9 years as well. And Lynn has published a great deal of the social and cognitive psychology around differential interpersonal experiences for the children. But when they got to 13, we sat down and we said, why don't we measure the cortisol? Now, I must hold my hand up here in case there's anyone who knows this story, because otherwise I'll be known for a fibber. I was the deepest skeptic, and I said, this is a complete waste of space. There's no chance that this study can give us individual differences in the way we're looking for. And it is so good to be proven to be so wrong. Here is the individual differences in cortisol at 13 between the children exposed and not exposed to postnatal depression. As you see, levels in cortisol are twice as high as in the exposed group. Now, there's been a series of smaller papers published since this biological psychiatry one, which point out, and I want to remind you, this is not a causal relationship between postnatal depression and this outcome. It's that the postnatal depression is indexing something that we don't really quite understand. It might be care, it might be something familial, it might be genetic. It's just that it's a marker of things going not quite right, I think would be the best way to put it. But we don't quite know what it is. And yet, here we are at 13. These individuals are not depressed. None of them have a current depression. These findings are all dealt with for the possible covariations and flaws in psychopathology. And then we follow them up 3 years later at 16, and Sarah, I think this has just come out, and we find that in fact at 16 years of age, the symptomatic group are more likely and this is now clinically symptomatic group, who don't meet DSM-4 criteria for major depression, but they are in this minor depression, probable depression kind of mixy group that don't do very well according to the longitudinal studies from Dunedin and Christchurch and so forth. They have significantly higher cortisol, and of course they were in the postnatal depressed sample. Now the secondary analysis suggests that we've got this kind of model going on, that can be no more than a post hoc possibility, which is that the maternal postnatal depression influences some kind of environmental set of events here that I've not marked. That produces the cortisol levels at 13 years, and it's the cortisol level at 13 years that increases the clinically meaningful depressive symptoms. So the mediation model doesn't hold. It's a sequential model that looks best for this, as it were. So here's a kind of messy attempt by us to take the Meany model and at least get one handle on it, which is the idea that early experience in a particular way is having effects on individual differences in cortisol, but it could be completely undermined if we knew what the genetics were. It could be entirely genetic. Maybe the whole thing is simply a series of epiphenomenal correlates of a gene-environment correlational model. It's the genes, stupid. Possibly there's an impact of early environment on the epigenetic program of the HPA axis. And the relative contribution of genetic factors on variations in cortisol levels in brain and periphery is simply not known. Now, we have looked at the glucocorticoid receptors and all their little SNPs, and it's extremely unhelpful because unlike the promoter, none of the frequencies are very large. So that means that we've got power problems of some size, and we're not going to get at it in this sample, or indeed in almost any of the samples we've got. We estimate you probably need a good 3,000 to 10,000 worth of individuals to get a really good answer. That's not impossible, but it's certainly not where we started with 91 kids. So I've taken you through now a bit about cortisol and the early environment. And just to remind you, I could just get you to recall that earlier on we were talking about serotonin having a role in neurodevelopment, being sensitive, producing brains that might be sensitive, or producing brains that might become sensitive as a consequence of events. But can these two things go together? Well, the animals data said of course they can. Serotonin and cortisol interactions have been around for ages. There's loads of stuff on animal work. But what about human work? Can we have chemical minds in concert? Would that begin to give us both the genetic and an environmental effects that would be underpinning the diathesis for when the brain is challenged that you're going to go in this direction or that direction, either before or after you've been depressed? So the primate studies show in rhesus, Steve Suomi's group in particular, that the S allele is not good news if you're deprived. You know, I'm sure there can't be any one of you in this room who hasn't seen Steve, as it were, perform these wonderful lectures, and if you haven't, you must get a ticket as soon as you possibly can, as he takes you through the history of his rhesus monkey colonies and shows you the remarkable things that they can do, showing the gene-environment interactions in his group, which also involve changes in cortisol. So we get closer, as it were, to our particular hierarchy of the mammalian system, not only in rodents but also in rhesus. And these have negative behavioural consequences. So as well as the chemistry, there are marked negative behavioural consequences, and I've seen Steve do this lots of times. He's got a wonderful slide of all the offspring who've been, peer-reared and family-reared, and the challenge is that he brings in a stranger into the large cage area, and he's got this on tape, and the groups completely segregate according to their experiences, and the fearful, shy monkeys that were S alleles and had poor upbringing right up in the top left-hand corner, and the L alleles had a good upbringing, and they're right up front saying, "Who the hell are you?" Down here, and in the middle are all the uncertain ones, a bit like British politics. How can we translate that kind of work into our work? Well, O'Hara from Stanford recently published what I think is the first study of using late life adults, looking for a main effect of genotypes on average working cortisol in community dwelling late life adults, humans. Now, this isn't a behavioural study. There is some very interesting neuroimaging in this study, but I just wanted to illustrate that here, as far as I know, is the first published gene by cortisol interaction, serotonin by cortisol. More evidence for this relationship between two chemical systems for which both genetic and early experiences are going to be important. Well, we've been looking at this, and here is this CHAMP study that Peter alluded to that is not published. And so I'm going to share some unpublished data with you, so you must take that as being, could be anything, could be good, could be absolutely no good at all. And it's a prospective study in which we measure all these things, and it's a high-risk study so that we can improve the yield of depressive episodes over a 12-month period, and we get about a 10% yield over a year, which is 3 times more than you would expect from a random sample. And just to start off with, sorry, let me just go back. So we looked in our sample to see if we can we find the O'Hara finding and all the animal finding between H-cert and our cortisol levels in this large sample of high-risk adolescents, and the answer is yes, we do. So here's a serotonin promoter by cortisol, morning awakening cortisol level, and you can see that the levels go up as you head down towards being homozygous, and this is true for both sexes. So that's not sex differentiated. It happens for both sexes, at least in our study. Now, recently, the promoter is coming under more and more chemical analysis, and somebody decided that they would find a SNP in the promoter, which was most unfortunate because the SNP changes the sensitivity. And Gil Saltzman and David Brent's group published this in the American Journal of Psychiatry. If you have the SNP, actually you're going to be more like an S, even if you look like an L, which is extremely unpleasant. So we, for the science, so we included the SNP, and it doesn't make any difference. You put the low efficiency SNP G group in with the S's, and you still get the same finding, and it's still sex differentiated. Still no sex difference. So the S allele is associated with increased levels of circulating cortisol across the lifespan. There's this O'Hara finding, there's our finding, there are others, but not in good human samples, and certainly there's conjecture. The first paper was actually published in 1997 on a sample of about 32 depressed individuals and another on 27, not long after Lesh first mentioned these points in Science. So is the liability for major depression in adolescents increased by the serotonin-cortisol relationship, or by one or both of these factors alone? And we can look at that in the CHAMP study because we've got the interaction, and we know who got depressed later, so now we want to say it isn't just cortisol that causes depression. There shouldn't be a main effects between HSERT and depression. There shouldn't really be a main effects between cortisol and depression. There might be, and we've certainly shown that there is in previous studies, but not when in a genetically sensitive design. And when you do that, you do indeed get a multiplicative interaction in which, if you carry one or both copies of the S allele, you're significantly more likely to be depressed, and you're significantly more likely to have high cortisol levels. But, as we were noting, about a third of depressed individuals who become depressed don't carry the S allele, and they don't have particularly high cortisol levels. It would be just as well, because depression is heterogeneous and has got lots of pathways. So to try and put all the mechanisms into one basket would be wrong, and I'm very glad that we don't have that finding. It's not all in one place. So the S by cortisol interaction is associated with subsequent onset of depressive episodes. What are the mediating effects between the genotype, the HP axis, and depression? Is this an example of epigenetic programming, or is this an example of major effects of genes on brain development, including the HP axis, and the environment is, as it were, getting in the way? I don't have an answer for that question. I obviously like the environment hypothesis, but that doesn't mean to say that I'll be right. We don't know, and we need much better studies than we've got on larger sample sizes dealing with gene-by-gene interactions, insofar as the power of those studies allows us to do that, and our knowledge base allows us to do that. What does this mean for the brain? Well, let me take you back to our little study, because we didn't do HSERT in this study, but we did have their adolescent cortisols. So we decided to look at hippocampal grey matter as adults and see if it correlated with their individual differences in cortisol 10 years earlier. And it does. It doesn't actually correlate with challenge cortisol. It doesn't correlate with changes in cortisol as adults. But 10 years on, with reduced grey matter volume, there is an inverse relationship with the level of cortisol when they were depressed as adolescents. It's a very small sample, and you could certainly get rid of this because there's a bit of a cluster there, and there's 3 guys, and this regression line could be eradicated within seconds. But it stood up to correction, which struck me as remarkable. So there's a possibility for some kind of ontogenic process that we're looking at in relation to the HP axis at one point in time and brain structure that might have been there at the beginning, so we can't actually argue about the direction of effects. Similar finding for increased hippocampal activity with something rather interesting, which is the cortisol levels again. So I threw that in because it seemed to us that we need to bring together physiology and genes, not necessarily the genetics of that physiological system, but the genetics as relevant to affective disorders, and the physiology that might be related to the way in which we deal with the experiences that we have, not just the emotional ones, not just the social ones, but internal ones too, and try and examine whether or not now in designs that allow us to image properly, measuring, for me anyway, measuring the social environment with a bit more precision than up till now, and incorporating perhaps studies of psychology that I've not talked about, but I'm going to suggest to you that this is the sort of map. So here's one of those really good things you can jot around on the plane and so on. Here's a map of the brain, two-dimensional flat map of the brain, of certain kinds of psychological systems and certain kinds of genetics. So we're going to say that the anterior cingulate and the prefrontal cortex are very important for conflict detection, and this is a summary slide from a whole range of studies by Sonia Bishop, for one, and others in the realm of neuropsychology, many others. Conflict detection and alerting control involve organisational components at the front of the brain. COMP seems rather important for this, and actually turning out to be quite important for those of you interested in psychosis and disintegrating stories of development, possibly personality, but I won't go there. Competition for processing resources becomes apparent as the top-down areas in the brain seek space, in inverted commas and as a metaphor, within neural systems, and coming up from the bottom, the bits that I've been focusing on, amygdala and hippocampal structures with their roles in threat detection, particularly at the level of the amygdala, and appraisal value, memory, and the appraisal value, the value of bringing things back, the value of encoding things in the hippocampal level, and these two things here being highly sensitive to serotonin mediation or moderation. I'm not quite sure which. Probably moderation if it's highly genetic. And appraisal value being cortisol moderation or mediation, probably mediation if it's a serotonin by cortisol interaction. But we would now get there by specifying the psychology a priori that we want to study. Instead of just kind of saying, fill in this questionnaire, I'm going to run a regression and see where you are on dimension, I'm actually going to test a specific hypothesis about particular components of psychology that might be the end of phenotype, that either is there because you may get depressed, we could do it that way, prospective, or you've been depressed and I want to see what's happened to you. I don't see why either of those things, as it were, would be wrong things to do, would be good things to do. In my view, anyway. This output here, by the way, refers to outputs that are about either the neural network output, that's why it's not an outcome, it's an output at the neural level, or it's an output in some processing level, something that one might measure, for example, like information processing speed or inhibition or so on. And finally, if this is the brain, and it's got, see, brains have numbers all over them these days, so perhaps yours does too. One of the important things is to disaggregate the social environment more. I think these have been fantastic studies by groups with the right data and have thought this through wonderfully, but I do worry about the lack of precision in the level of the social analysis, and I think we need to think about that social analysis a bit better now in future studies which allow us to disaggregate what we mean by maltreatment in relation to friendship development, in relation to major life events, in relation to chronic family discord, and so on, in ways that certainly are possible from existing data sets, hopefully from future ones. What I wanted to do, and you'll be the judge of how successful it's been, was to walk us around how we go from a longitudinal studies of individuals with individual differences in outcome of their clinical states to thinking about the substrate on which we're working, which is the brain, but its substrate is its genetic formation. But how might those genes be, as it were, open to epigenesis? And my hunch is that they are, and I hope they are, because I think that gives us a much better brighter future for interventions, both at different points in the life course. Because what I haven't told you is that we don't just have immutable effects of these things in infancy. There are changes in the life course that might well allow us to do much more than we've done so far, but also for us to think. I could bring you all the way back to my little anecdote in a small group about persistent disorders who were subject to very, very poor outcomes which seem to be correlating with substance misuse, is that we'll be able to provide much better public advice by knowing where are we going to aim our our intervention strategies in terms of prevention and promotion, of what kinds of brains, if I can put it that way, as well as within individuals, we're going to be very concerned about. I'm going to stop there. Thank you very much for listening to me.
Well, that was fantastic. Yeah, that was fantastic. You made me feel, even if briefly, that actually the possibility of understanding depression was at least within your grasp, if not within mine. It's a remarkable integration of different levels of analysis, and what I think is totally remarkable about it is how Ian can bring together the psychosocial with the biological, both in terms of guiding treatment and prevention. We do have some time for questions, particularly questions of clarification. And there's a mic that's going around, so if you can try and draw attention to yourself. Question up there. Could you introduce yourself to us, please?
Martine Flamand. It was a fascinating lecture. You said that it's very important to have reliable measures of the environment. So what would you recommend as reliable measures?
Okay, thank you, Martine. Well, if we take the very genetic position that the environment is everything that's not genetic, then in a sense you need multi-levels of reliable measurement. So, for example, if we're going to do physiological measures, then we have to make sure that we undertake them in a robust manner. If we do cortisol measures, as you know, but some of you might not, you can't take a single measure. In order to get reliability, you have to deal with individual differences over days and time. So you have to put in the spade work to produce a robust cortisol measure, for example. Also, from now on, I think it's clear we're going to have to dissect that kind of measure into potential trait-like phenomena and potential state-like phenomena. So we're going to have to undertake, for example, latent trait analysis to dissect those features as well. If I was going to do a social measure, well, I still think that the measures that we used in the 80s, that we based on George Brown and Jean Paykel's work, are the best ways for measuring social experiences. I mean, social experiences has a limited reliability framework. I mean, the minute you say to someone, I'm going to ask you about what's happened in your life recently, you know as well as I do, you're not going to get a fully correct answer. If I was to ask you what you had for breakfast on last Wednesday, it might be a struggle for you to tell me exactly what that was. So when we give out questionnaires for life events and difficulties, we're inviting trouble with reliability. You can make them reliable, that's no question, but then you're really squeezing the validity out of the enterprise. And that's my worry about some of the studies so far, which have said we've got great genes, we've got 250 people, we took this life event measure and we just measured a few life events and we got ourselves a relationship. I think that's a problem, and I think Ken Kendler showed that in his twin study, where he did a very, very comprehensive measure of both major and minor life events, as well as his genetics, as well as his twin data, and got a slightly different result. Well, that slightly different result might mean all the difference between understanding quite what that relationship is and what it isn't. For example, Ken's study also shows a much stronger relationship with the homozygosity for the serotonin promoter and milder life events. No one else has so far replicated that, to the best of my knowledge. If anyone knows, I apologise, but I think that that's the kind of thing where you've got to get that measurement right. If you're specify the question, find the best environmental measure. For example, in the archives last year, Jim Van Oz said, I think, and his group said, I think the intermediate measurement, which I haven't talked about at all, is personality, temperament, some kind of cognitive measure. So they specified a cognitive measure, and they looked at neuroticism as their cognitive measure. They made sure they had a very robust framework for the measurement of neuroticism, and they showed a gene by environment interaction between neuroticism, and this is summarising an extremely complicated paper in my view. I didn't find it easy reading, showing that the gene by neuroticism relationship might be the one by which the gene by life events measure is mediated. So that's what you have to do if you're going to do studies that involve communities. If you're going to do experiments in the scanner, the issues are just the same. You must make sure that your experiments are robust. For example, if you're going to do a memory task, do it offline and online. Do it more than once. Use different memory tasks that test the same construct, so that when you actually get to the scanning, precious scanning time, you can interpret changes in bold, if it's it's obviously a functional task, with a degree of reliability that isn't just going to rely on a great deal of SPM2 post-analysis, which is always, I mean, bioinformatics is terrific, but it really would be good to have robust experimental tasks. And I think there is a tendency at the moment for very, very clever people saying, I've got a new task, I want to use my new task. And I think that should be just as reliable as the environmental measures in longitudinal studies. And of course, a pseudo gold standard, as it were, would be, there's the cohort community study, sequential longitudinal design, robust measures of cognition and life events, and now I'm going to do neuropsychological studies of individuals who've been depressed, individuals who've never been depressed, individuals at risk but yet to be depressed, individuals who've recovered and shown resilience, and so on, and I'm going to do the experiment on that, embedded in that longitudinal cohort. That for me would be as the limits of knowledge so far. If you then add asymmetric genetic analysis and you've got about 50 or 60,000 people in the study, you might have an answer.
Is that the next grant application? Yeah.
We just got that grant.
Oh, right.
No, it's not true.
Any further questions?
I think you've all done very well staying this long, if I may say so. If
there are no further questions, I want to thank Ian again for an absolutely brilliant lecture. Ian, fantastic.